Hello, and welcome back to Neuroreview. In this episode, we're continuing our dive into the neuron. In the last video we explored the soma of a neuron, which is encapsulated by the membrane, contains organelles, and is supported by the cytoskeleton. While these structures are vital to the survival and proper functioning of a neuron, they are not unique to neurons alone. All cells in the body have these structures. In this video, we'll take a look at one of the types of neurites, which are the projections from the soma. In contrast to the parts mentioned earlier, these neurites are unique to neurons. Of the two major categories, we'll start with a neuron's axon. The role of the axon is to transmit information to other parts of the body through an electrical signal called a nerve impulse. This could include different neurons, muscles, or glands, depending on what the other end of the axon is connecting to, or innervating. The principal axon projection can extend from less than a millimeter in some neurons to over a meter in length in others. In terms of diameter, axons are typically 1 to 25 micrometers across in humans, but can be up to 1 millimeter in some squid. That's up to 1,000 times larger. As we'll see in a future video, a large diameter axon is helpful when studying neurons in a laboratory, but is also related to the speed at which information travels down the axon. The region where the soma of the neuron leads into the axon is called the axon hillock, which is characterized by a tapered segment. Starting here, as well as further down the axon, there is no rough endoplasmic reticulum or ribosomes. Additionally, the axon membrane has a different protein profile than the soma membrane. Let's examine these distinctions for a minute. Axons lack rough ER and ribosomes, which are the machinery that produce proteins. This means there is no protein synthesis in the axon, which means that proteins present in the axon must have originated from the soma. If these proteins are the key difference between the axon and the soma, then these axonal proteins are likely the source of the axon's information-sending properties. The middle part of the axon is called the axon proper. Here, axons can branch, forming axon collaterals, which can innervate other parts of the nervous system, or even loop back to innervate the neuron they came from. The end of the axon is called the axon terminal. This is where axons come into contact with other cells, which may themselves be neurons. The point at which the axon terminal connects with the other cell is known as the synapse. The axon terminal has a few characteristics of note. Microtubules do not extend into the terminal, and the terminal contains many small membrane bubbles, called synaptic vesicles. The inside surface of the membrane that faces the synapse has a dense covering of proteins, and the axon terminal contains many mitochondria, which indicate a high demand for energy. What is in these synaptic vesicles, and how do they let a neuron communicate across a synapse? Since there is no protein synthesis in the axon, how do proteins get to the axon terminal? What structure receives the signal an axon sends across a synapse, and how does this structure, along with axons, help us classify neurons? This will be the topic of the next video. To review, in this video we explored the various components of the axon of a neuron. The axon, which can't make proteins, sends nerve impulses from the axon hillock, through the axon proper, to the axon terminal, where the axon contacts another cell, making a synapse. Thank you for watching this video. If you liked it, make sure to like, share, and subscribe. As always, if you have a suggestion for a topic or a note on something that could be improved in a future video, please leave a comment down below. Thanks for stopping by, and I'll see you in the next video.